ladies and gentlemen. So now I want to start the Symposium 2. And my name is Shinichi Kosaka, the Director General of the National Institute of Neuroscience, and I will take chair of the sessions. The Symposium 2 is research development in relation to brain, mind, and society from the viewpoint of biological research. Okay? And the first speaker is the Professor Ian Everall, and he is now the Cato Chair of Psychiatry, University of Melbourne. And he has a lot of research careers. I want to skip all of his careers. Okay? And his research interest in these 20 years is the molecular, cellular, and genetic changes in the brain in major psychiatric disorders. And he also recently, his interest is the psychosis, major depression, and bipolar disorder and cognitive impairment, especially when associated with the HIV infection and methamphetamine treatment. So today's title of his presentation is a very big one. Okay? Neuroscience and psychiatry. So,
I'm honoured both for the invitation to speak to you today and also for the deepening and growing collaboration between the National Centre for Neurology and Psychiatry in Tokyo and the Department of Psychiatry at the University of Melbourne. Today I'm going to talk briefly about the relation of neuroscience research and psychiatry and society. I want to do four things. I want to first of all make a statement of why we undertake neuroscience research, which is we're trying to help patients, people affected by mental illness. I then want to talk about why this area of research is so important. I'm then going to give two examples of current neuroscience research activity that's occurring in the Department of Psychiatry at the University of Melbourne. And then I'm going to finish by suggesting how we could possibly do neuroscience research a little bit differently in the future. We carry out this research in order to improve the quality of life of our patients. And of course, the ultimate aim is to return our patients to health. And we heard this morning the definition by the World Health Organization of health. Why is this area of research so important? It's important because it affects so many people and the economic costs are so large. The Society for Neuroscience in the United States said that in 2007 there were at least 1 billion people affected worldwide by neurological problems, and that these diseases account for 11% of the world's disease burden, and this does not include mental health problems or addictions. The European Brain Council stated that in 2010, neurological disorders such as stroke, Parkinson's disease, Alzheimer's disease, epilepsy, etc., that in Europe cost $1 trillion a year. These are not trivial amounts of money. Mental illness itself is again very expensive. We heard earlier on that as a cause of burden, major depression, which is currently the third largest cause of burden in the world, will move to number 1 by the year 2030. And if we look at the cost of major depression, again, they are very high. There are direct costs, the cost of involving mental health services, general practitioners, taking medication, being admitted to hospital, and these costs are over €500,000 in the United Kingdom and over €2 billion in the United States. And then there are indirect costs, and these are often related to the inability to work properly and the loss of productivity. And these costs are even higher. over 11 billion euros and another 4 billion euros from early mortality, often through suicide. So these are extremely important disorders that we need to understand more and have better interventions and better outcomes. If we look at the loss in productivity, here's a study that looked at the number of working days lost in people with major depression in 4 European countries. And you can see that compared to the individuals who are not depressed, there's a striking increase in the number of working days lost. And if we compare people with no depressive symptoms to those with acute depression or chronic depression, again we see a significant increase in absenteeism, that's missed work days, as well as presenteeism. And this is where people go to work while they're depressed, but do not work as effectively. And if we look at this a little bit more, we see that people with moderate or severe depression, or even very severe depression, have significant increases in their inability to work. They get less work done, the quality of the work is poorer, and they make more mistakes. For society, this has significant implications on productivity and economics. So I hope I've convinced you that mental health problems or mental illness are very important problems for all countries. And now I want to move on to my second point, which is to give you 2 examples of neuroscience research in the departments of psychiatry. The first example is work being carried out by Professor Brian Dean, Associate Professor Elizabeth Scarr, and myself, trying to understand more about the molecular pathology of major psychiatric disorders, schizophrenia, bipolar disorder, and major depressive disorder. After that, I'm going to give a second example of work carried out by Professor Stan Skafidis, Professor Christos Pantelis, and myself in developing a diagnostic test for autism. With Brian Dean and Liz Scarr, we were looking at the role of inflammation in psychiatric disorders. And here is a summary table of 20 inflammation-related proteins being studied in a number of studies. And a major depressive disorder, there are 32 studies saying that these inflammation-related proteins are increased, 36 saying no change, and 7 saying they're decreased. And a similar number, a smaller but similar set of findings in bipolar disorder and schizophrenia. However, it's difficult to interpret variations in inflammation-related proteins because they can vary due to sleep disturbance, change in body weight, glucose status, variation throughout the day, and even technical sample storage conditions. But despite this, we thought it was worthwhile investigating inflammation-related proteins in psychiatric disorders. We decided to look at the expression of all genes in the human genome in regions of the brain. So we took brain tissue and we extracted the RNA, hybridized it to a gene chip, and then from the data that emerges on how all genes are expressed, used 2 software programs to analyze the data to tell us what gene expression has been changed. I'm not going to go through all the details because there's 39,000 genes, but one gene in particular was significantly changed, and that's this inflammation-related protein, tumor necrosis factor alpha. So I'm going to concentrate on this as an example. There have been a number of studies in psychiatry showing that tumor necrosis factor alpha in the blood is increased in major depressive disorder, a few studies suggesting it's increased in bipolar disorder, and a couple, maybe one, suggesting it's increased in schizophrenia. Before I present the results of this, I'm going to explain the biology of tumour necrosis factor signalling, because it's quite complicated. So here we have tumour necrosis factor, the protein, bound to the membrane of the cell. And it can interact with two tumour necrosis factor receptors, receptor 1 and receptor 2, on other cells. And when it's bound to the membrane, it prefers to interact with receptor 2. However, this protein can cleave tumour necrosis factor so it becomes soluble. And when it's soluble, it prefers to interact with receptor 1. To make matters more complicated, this protein can cleave the receptors, making them soluble and interacting back with tumour necrosis factor in the membrane. So you can have signalling both forwards and signalling backwards. So this just shows examples of the type of signalling you can have. You can have non-cell contact going forwards or backwards, cell-to-cell contact going forward signalling and backward signalling, etc. So what did we find? We carried out the study on the dorsolateral prefrontal cortex, which is implicated in schizophrenia. Thinking of these various components of TNF signalling, the membrane, the soluble, and the two receptors, in major depression, we found a highly significant increase in the expression of the TNF, which is bound to the membrane, and a small increase in the gene expression of receptor 2. By contrast, in bipolar disorder, there was no change in the transmembrane TNF, but there was an increase in the gene expression of receptor 2. And in schizophrenia, there was no change in the transmembrane TNF, but there was a large increase in the gene expression of receptor 1. So what do these findings mean? Here's the complicated signaling and the various molecules involved. From our findings, we think that major depression is an abnormality confined to the transmembrane TNF-alpha. In bipolar disorder, it's an abnormality of the receptor 2, whereas in schizophrenia, it's an abnormality of receptor 1. We're still working on this and still trying to understand the implications of this for the various disorders. I now want to turn to the second example of research activity in the department, and this is trying to develop a diagnostic test for autism spectrum disorder. Here we wanted to look at the natural variations in genes that occur across the human genome, and what are called single nucleotide polymorphisms, where a nucleotide changes, say, from a G to a T. There are about 1.5 million of these variations across the whole of the human genome. And we were able to get the data sets of these gene variations, or SNPs, from an autism genome research exchange. We had access to almost 7,000 individuals where had their complete human genome variations, called GWAS, Genome-Wide Association Studies, of which 2,500 had autism spectrum disorder. We fed the information into a computer and, using various software programs, trained it to try and look at those variations to see if it could tell the difference between individuals with autism and individuals without. And eventually, after lots of training, it was able to find that using just over 200 SNPs, we could accurately differentiate with an accuracy of 85% whether someone was autistic or not. So here's a diagram showing the distribution of the control individuals and the distribution of the autistic individuals. And you can see there's not much overlap. So this is the first time that we've had a diagnostic test to help with the diagnosis of a psychiatric disorder. Also in the data set were quite a few parents of individuals with autism spectrum disorder. And interestingly enough, the parents in green fell directly in the middle between the controls and the autistic subjects. And we also had access to an independent data set, and again, our classifier had an accuracy of 74% in diagnosing whether someone was autistic or not. So I hope from these two examples I've indicated how neuroscience research can help our patients or those suffering from mental health problems. The first example relates to experiments trying to understand the molecular and cellular pathology in mental illness, and the second one using variants in genes to develop a classifier or diagnostic test. But is there anything we could do somewhat differently in the future to help our patients even more? And I think there is. We tend to have a rather pessimistic view of psychiatric illnesses, especially psychosis and schizophrenia. And I think our view is coloured by the original descriptions by Emil Kraepelin and Eugene Bleuler, who both emphasise the chronic nature of these disorders and the poor outcome or recovery. However, as Bleuler's son, Manfred, showed in a hospital study, over 60% of those on their first admission had recovered significantly, and 20% had recovered fully. Similarly, Ming Swang in the Iowa 500 study showed that 46% of individuals with schizophrenia had either improved or recovered. So we should think about these disorders more optimistically, and that we could actually help our patients have a better quality of life. As we heard from Carol Harvey this morning, those individuals living with mental health issues have different priorities to ourselves. We think in terms of diagnosis, and we think in terms of symptom reduction as being the most important things. And we often carry out our neuroscience research with those symptoms and those diagnoses in mind. And as Carol said this morning, while symptoms such as depressed mood and hallucinations can be distressing, patients are more interested in issues pertinent to the recovery model of mental health, and these relate to their own level of functioning in society. And so there's been the development recently of the recovery model, and this is a social movement influencing mental health service development. And it refers to the subjective experience of optimism about outcome from psychosis and other mental disorders, with a belief of the value of people suffering with these disorders and their empowerment. And the services are now focusing on which decisions about treatment are taken collaboratively with the user, and helping the patient or user find productive roles for themselves in society. So I think we could do our neuroscience research better if we related our findings to those issues which are important to those living with mental illness. And these issues, and therefore these markers, would be around employment, relationships, cognition, so they're functioning in everyday life. rather than their symptom severity. I think this is a new way of thinking for neuroscience. I think it's going to be challenging, but I think it may mean that our research means more in the real world to the real people we're looking after. Thank you very much.